Okay, ladies and gentlemen, our opening ceremony is about to begin. Okay, bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Mr. Roger, and absolutely good afternoon for everyone here. Welcome to webinar Fabi Unsud. We proudly present the second series webinar on Aedes aegypti and Aedes albopictus, diversity, phylogeny, and phylogeography, Faculty of Biology, General Sudirman University. Okay, at this moment, I would like to say welcome to Professor Roger Frutos, who will be the speaker at our webinar today, and the Excellency Dr. Dwi Nugroho Wibowo MS, Dean of the Faculty of Biology, General Sudirman University. And the committee of this event, and absolutely welcome to all the webinar participants who attend today. Okay, first of all, let's say thanks to God who has given us guidance, happiness, health, and mercy so that we can attend and participate in this evening without any obstacle. Okay, ladies and gentlemen, the webinar on Aedes aegypti and Aedes albopictus, diversity, phylogeny, and phylogeography is to celebrate the 59th anniversary of the Faculty of Biology, General Sudirman University. And thank you so much for everyone here for making time to attend this remarkable webinar. Various institutions attend this event. There are 93 institutions registered in this event. It's so unpredictable for us. And for this event today, we have some agenda. And so let me read some our agenda for this event. The first is the opening. The second is singing Indonesia Raya. The third is welcoming speech. And the fourth, the core event webinar. And the last is closing. Okay, before we start our agenda, I will read the webinar rules for this event. The first, participant use username or name ID with full name _ institution or location. And then during the event, participant microphone are deactivated and can deactivate with the permission of the committee. And the third is question related to the material can be submitted via Zoom chat with the format name _ institution _ question. And participants are not allowed to express opinion or other information that often Sarah or is a political issue. And participants make a presence on the form that the committee will provide at the end of the event. And the last e-certificate and material will be submitted via a link that will be distributed by the committee via email to each participant according to the data received at the time of attendance. Okay, before the event continue, let's take some picture for documentation. Please, everyone here can open your camera for this moment. Okay, Mr. Eko, you can take some picture. Okay, 1, 2, 3. Okay, once more again. Okay, Mr. Eko. 1, 2, 3. Okay, thank you so much for everyone here. Okay, ladies and gentlemen, let's open the webinar by reciting Basmalah. Bismillahirrahmanirrahim. Okay, the next agenda is singing Indonesia Raya. Okay, the following agenda will be a welcoming speech. Allow us to invite Dr. Dwi Nugroho Wibowo, MS, as a Dean of the Faculty of Biology, Jenderal Sudirman University, to deliver his welcome remark and continue to open the webinar. Please welcome Dr. Dwi Nugroho Wibowo, MS.
Masih, Mas. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, good afternoon, sejahtera bagi kita semua. Yang saya hormati Wakil Dekan Bidang Akademik, Dr. Mono, Wakil Dekan Bangun dan Keuangan, Dr. Heri Pratikno. Saya hormati Koordinator Program Studi Biologi Program Doktor, Prof. Agus Nurjanto, Koordinator Program Studi Biologi Program Magister, Dr. Lestari, kemudian Koordinator Program Studi Biologi Program Sarjana, Dian Bhagawati, dan Koordinator Program Biologi Program Budidaya Ikan, Randa Sugiarto, MSc. The Honorable Prof. Roger Frutos from Sirat, UMR 17 Intertrip, earlier from good afternoon or good morning in French. Prof. Roger, nice to meet you. On behalf of the Faculty Biology, I would like to welcome all of you to this webinar series about Aedes aegypti and Aedes albopictus, diversity, phylogeny, and phylogeography. Especially, I would like to express my appreciation to Prof. Roger Frutos for his time to share knowledge to us. We hope you enjoy for having us along this webinar. In this webinar, we would learn about the diversity, phylogeny, and phylogeography of Aedes aegypti and Aedes albopictus from Prof. Roger directly. Professor Roger, I'm sorry and permission, I will use Indonesian. Yang saya cintai dan saya banggakan para peserta webinar seri 2 yang berbahagia, serta seluruh hadirin yang saya muliakan. Pertama-tama, marilah kita panjatkan bersyukur kepada Allah Subhanahu wa Ta'ala, Tuhan yang Maha Kuasa, karena atas rahmat dan karunia-Nya pada siang hari ini kita dapat hadir secara daring dalam rangkaian acara memperingati Dies Natalis ke-59 Fakultas Biologi kita, Fakultas Biologi Unsur ini, dengan menyelenggarakan webinar series 2. Kesempatan ini adalah series 2, kemarin sudah diawali webinar seri 1 dan nanti akan dilanjutkan seri berikutnya. Pada webinar series 2 ini, topiknya adalah Aedes aegypti dan Aedes albopictus, diversity, phylogeny, and phylogeography. Dan narasumber yang tadi kami sampaikan adalah Prof. Roger Futos, beliau dari SIRAT UMR 17, Intertrip Montpellier dari Prancis. Ibu Bapak yang saya hormati, para peserta webinar series kedua yang berbahagia ini, pada kesempatan ini sekali lagi saya sampaikan terima kasih kepada Prof. Roger yang telah berkenan berkontribusi dan bersinergi dalam pelaksanaan webinar kali ini. Terima kasih juga kami sampaikan kepada Prof. Indang Sri Murni Kusmintarsih yang telah berkenan berkorespondensi, berkomunikasi dengan Prof. Roger sehingga salah satu rangkaian webinar ini bisa terselenggara. Dan kami, Fakultas, sangat mendukung dan sangat bangga, kemudian juga mengapresiasi kegiatan ini, sehingga apapun yang diperlukan, yang dibutuhkan di dalam kaitannya kegiatan seminar ini akan kami support full sepenuhnya. Terima kasih juga kami sampaikan kepada para panitia webinar series kedua ini, serta Ibu Bapak semuanya yang hadir pada kesempatan siang hari ini atas kontribusinya, atas kesediaannya, sehingga nanti bisa mengikuti dan insya Allah nanti kita akan dapat banyak ilmu, banyak pengetahuan dari Prof. Roger. Dan harapan kami semoga acara ini bisa berjalan dengan lancar dan diperoleh hasil sesuai dengan yang ditargetkan, serta semuanya selalu dalam lindungan Allah Subhanahu wa Ta'ala, Tuhan Yang Maha Esa, dan semuanya selalu dikarunia kesehatan. Ibu Bapak yang saya hormati, tentu tidak bijaksana kalau saya terlalu banyak menyampaikan sambutan pada siang hari ini, dan izinkanlah Ibu Bapak yang saya hormati dengan memohon ridho Allah Subhanahu wa Ta'ala seraya mengucap Bismillahirrahmanirrahim. Webinar seri 2 Aedes aegypti dan Aedes albopictus, diversity, phylogeny, and phylogeography yang dilaksanakan oleh Fakultas Biologi Unsur Purwokerto pada hari ini, Rabu 3 Agustus 2022, secara resmi saya nyatakan. Terima kasih, maju terus pantang mundur, kenal menyerah. Bila taufik wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much, Dr. Dwi Nugroho MS, for the welcoming speech. Okay, ladies and gentlemen, now we come to our main agenda. The presentation will be delivered by Prof. Roger Frutos and also will be guided by Professor Endang Srimurni Kusmintarsih, PhD, as a moderator. Please welcome Prof. Endang Srimurni Kusmintarsih. Time is yours.
Okay. Thank you, Mas Trisno, for giving me the time to be a moderator. Yeah, I would like to say good morning, Prof. Roger, and good evening in Indonesia. Thank you very much for being willing to be a speaker in this seminar, and also welcome to the Faculty of Biology, Unsur. Thank you also address to all participants consisting of a researcher, lecturer, and student for joining this webinar. Even I see that the participant, it is come from Africa, especially from Sudan. Yeah, Dr. Muhammad, welcome and thank you for joining in this seminar. And before starting, I would like to read the curriculum vitae of Professor Roger Frutos, and participant can follow through this slide that's come up. Just a second. I would share the PPT. Do you got my PPT?
Not yet.
Not yet? Hopefully will coming soon. I think already.
Yes.
In my other computer already come up. Not yet in yours?
Done, bro.
Okay. Oh, yes. Thank you. So I would like to read about the curriculum vitae of Professor Roger Brutus. Start from personal information that the first name and surname Roger Brutus and nationality French, date of birth 27 of March 1961, desired employment, occupational field as a professor or research director. About education. Prof. Roger passed from MSc in 1984 and passed from PhD in 1988. In the subject principal is parasitology, pathology, and name providing education in University of Montpellier II University, Montpellier, France. Continue to work experience. For working experience, it is consists in several periods, and I would not read all because many, and then will take time for reading all of the work experience. It may be just one or two periods. Work experience start from 2018 to now and the future. Yeah. Occupation or position held as a researcher director or professor, and then director for development at Association of Southeast Asian Institution of Higher Learning, or ASAIHL. Invited professor at the Faculty of Medicine Ramati Bodhi Hospital at Mahidol University, Thailand. Invited professor at the Faculty of Medicine at University Gadjah Mada, Indonesia, and former Deputy Vice President for International Relations at University of Montpellier. Main activity and responsibility as a dynamic of emerging transmissible disease, R&D on novel generation of surveillance and diagnosis tools, coordination of the international master dynamic, and project development at Aedes. Experience in edition. Former edition at Infection, Genetic and Evolution by Elsevier. Also editorial board member at Infectious Disease of Poverty in MBC. Editorial board member at Viruses in MDPI. Editorial board member at Vaccine, MDPI. Editorial board member at COVID in MDPI. Editorial board member at Microorganism in MDPI. Guest editor at Frontier in Medicine and guest editor at Frontier in Public Health. So maybe Bapak Ibu and Saudara will send the manuscript to this journal, will paste to Professor Roger. This is the experience start 2018. And next slide, this is start from 13 to 18. I would not read all. And then this slide start from 2006 to 2012. You can read from here. And then still many more, but I didn't read all, just like this. And this is the first, yeah. Start from 1980 to 1991, occupation as postdoctoral fellow, and then main activity and responsibility postdoctoral fellow at the Department of Entomology, characterization of Bacillus thuringiensis toxin chains, development and recombinant strain of Bacillus thuringiensis and baculoviruses, and name and address of employer University of California at Riverside, CA, United States. And then the last about his publication. Of course, I would not read one by one because a lot, yeah, until 296. So if I read all, then will take maybe half an hour. Yeah. There are 296 publications. Okay. I think like that, what I can perform for you about the curriculum vitae of Professor Roger. I apologize, Prof. Roger, I didn't read all of your curriculum vitae, but I would share in the in our faculty maybe later. Okay. And now we are going to the main session, the main program is presentation from Professor Roger. So the time is for you, Professor Roger.
Thank you very much, Professor Endang. So first of all, please let me thank you for the invitation to participate to this seminar and give this presentation. So I would like very much to thank the Universitas General Sudirman, but in particular within the university, the Faculty of Biology and the Professor Dwi Nugroho Wibowo for this kind invitation, and in particular, Professor Endang for inviting me to give this presentation and for giving this very nice introduction of myself. So thank you very much for the invitation, for the introduction, and for giving me the opportunity to discuss with you a quite interesting issue on a problem which is a worldwide problem, the problem caused by mosquitoes, Aedes aegypti and albopictus, and of course the disease they can carry. So to start, I will try to share my screen. Please just to tell me if you can see the slides.
Yeah.
Okay, very good. So I'm going to make the larger screen if it works. I hope so. Voilà. And just one last question before starting. Can you see the slides moving?
Yeah.
Okay, lovely, very good. Thank you very much. So, so let me start this presentation. So as mentioned during the introduction, so I'm going to talk about Aedes aegypti and Aedes albopictus, and mostly about diversity, phylogeny, and phylogeography, which are things I believe many of you are familiar with, and the things which are done very frequently all around the world, actually. So the first thing to do would be actually to start with some definition, because I say phylogeny, phylogeography, genotyping, but we need, before going into the heart of the topic itself, to give some definition and really narrow down and frame well what we are talking about and what we are doing. So let's go first, if you allow me, with some definition. First, phylogeny. Okay, what is phylogeny? Well, phylogeny is simply just by its name, because it's made from initial Greek words, like many words in science. So philo, well, comes from an ancient Greek term, phylon, which means raised tribe. Also, you have the word phylum going from that. Okay, and the geni, which come from the Greek genea, which means the origin. you find the same root in the word genesis, which means born. So what is the phylogeny? Basically is the analysis of the origin or the manner, the way of development. It covers both aspects. That is phylogeny. So let's go a little bit more into that. What is the, what do we do when we do phylogenetic analysis? It works on mutations, first of all. And we're going to analyze the mutations, not all mutations. We are going to analyze only the substitutions. In other words, when one letter, for instance, that one here, A has replaced a G, okay? That's phylogeny is working on exclusively on that. Phylogeny does not take into account the indels or insertions, deletions. When, for instance, a base is missing, like G here, or there is an additional letter, base here, like a G. So this is not considered at all. And it's also a problem because, of course, phylogeny doesn't consider that, but these indels are extremely important in terms of evolution, adaptation, and when you see indels is very often, not always, but quite often, an indication of adaptation to something. Okay, the protein was changed. So it's very important to consider that from the very beginning. So we are working only on a specific set of modifications, the substitution only, which means that when we align sequences and when we analyze this data, we work only, exclusively, only on regions which don't carry any indel. If there is an indel, this region is eliminated automatically by the software, by the program. You don't even see it, but it doesn't consider it. So another point is that phylogenies are based on characters. Okay, a character could be any attribute of an organism relating to his history. So we're analyzing the ancestry. Here we're talking about molecular phylogenies. So therefore, the characters, as we said before, are nucleotides, and more specifically, nucleotide position in a gene sequence with 4 possible character states, in that case, A, C, G, or T. That's our basic system, and we're going to work on the ancestry. That's very important. So just give you some word, again, some definition. In phylogeny, the way the characters are characterized, you have different way and different specific definitions for that. For instance, the plesiomorphy, which is the ancestral character state, the way it was at the origin. Apomorphy is when the character state is different from that of the initial one. So it's a derived state. You have the synapomorphy, when the derived character state is shared by one or several taxa or species, for instance, due to inheritance from a common ancestor. And you have the autapomorphy, which is something absolutely unique. It does not exist anywhere else, just in one place. Okay, the homoplasy, which is the identical characters obtained through not inheritance from the same ancestor, but independently from different ancestors, which are nothing in common. This is what usually we call a converging evolution or convergence, parallelism, or reversion, you see? So in phylogeny, you have different things that can be shown, can be illustrated by different pathways, to see how the character moved and evolved based on the ancestral one. So we have to make also difference between homology and homoplasy, because there is very often in confusion. A character state shared between 2 DNA sequences or taxa may be so because it is inherited comes from a common ancestor, then it's called homology, or it's independent evolution, then it's called homoplasy. So now I'm going to make a very important remark, and I'm going to stop here with this definition of phylogeny, because what I wanted to show you by these few slides is that basically nobody is doing that. Except for a very few number of people, basically nobody is doing phylogeny or phylogenetic analysis, mosquitoes or other things. The vast majority of people, including myself, we are not doing phylogeny. We're not doing what I mentioned earlier. We call it phylogeny, but it's not true. We're not doing phylogeny. What we are doing is diversity analysis and genotyping. We use the phylogenetic tools and the software, but we don't do phylogeny. And this is leading somewhere to some confusing situations, because there may be some mistake or some misinterpretations, because we sometimes try to make the results say something they cannot say, because they are not used in the right framework, where they are not used for phylogeny. So the main point in this definition is that I would say basically almost nobody, again, with the exception of very few people, almost nobody is really doing phylogeny. We are doing diversity and genotyping. So, but we are using the tools. And one tool we are using, which is very important, is the phylogenetic tree. Okay, what is a tree? What is a phylogenetic tree? It's not something magical, and is not the answer. Whatever you do, whatever analysis you do, you will always end up with a tree. That is no problem, because it's just the visualization. The problem is it relevant? Is it significant? That is the real question. So it's just a mathematical object, a graph, and you will always end up with a tree. The problem is that not all the trees we use are phylogenetic, first point, okay? And these phylogenetic trees represent only and exclusively the object which is studied, so the gene or the sequence we are working on, okay? And it represents only the phylogeny, if it is a phylogenetic tree, it represents only the phylogeny of that gene and not of the whole species. So when you see, for instance, we say phylogeny of Aedes aegypti, it's wrong. It is completely wrong. It's the phylogeny of this gene from Aedes aegypti. And if you do the phylogeny on another gene, then you may find something completely different, because each gene has its own history of inheritance, movement, etc. And the phylogeny of the global phylogeny of the global one species, it does not exist. It's because it's a mixture of many things. So it's the phylogeny for individual sequences, individual genes. So that's why it is important to see and to state clearly on what we are working. So just to conclude on that part, on the, I would say on some definition of phylogeny and the use of the tools we can make, basically we are using two kinds of tools, two kinds of trees. And when we build trees, 99% of the time, we have two approaches. One is the neighbor joining, NJ, and this is something that is extremely fast and easy to do, okay? But it's good only for working documents and quick analysis, and because it's not completely reliable. Usually we work it with a limited number of bootstrap, 100, 300, but it's very important to always have a group to root the system. You have to keep in mind that the NG approach is providing an unrooted tree. Even if you provide an ad group, it is by definition an unrooted tree. So it's not good for phylogeny, it's not fully reliable to analyze accurately the relationships between different sequences. However, it is a very good tool to go quickly when you have an idea of what is going on, because it's fast, it doesn't take much time to do it, a few minutes is done, etc. Whenever we want to do something very good, accurate, rigorous for communication, for publication, something very good, then you've got to run a maximum likelihood, ML, and which is attached to a specific evolutionary model. The evolutionary model will vary depending on the gene you are using. So you have to calculate that also. The problem with ML is that it is very accurate, it is very rigorous, but it is very long and very power consuming. For instance, at the very end of this talk, I'm going to show you some very recent results. Very recent, actually, we are working on it. I got the last tree yesterday. So, for instance, to build the tree, the ML tree on the COX-1 gene of Aedes aegypti, it took us 3 days and a half to run that tree I'm going to show you. So it could be extremely long, but it's very good. So, but you always, with Bootstrap, of course, preferably 500 or 1,000, so a lot of bootstraps to increase the statistical significance. And of course, always with an out-group root. And this time, the out-group is very important and very accurate because these trees, ML, are really rooted. So the out-group is really significant. So I'm going to stop on this overview here of the methods, I would say, the phylogeny, because as I said before, we don't do phylogeny. What we do actually, most of the time, is genotyping. So same origin of geno, like phylogeny, so the genes, the born, the origin, and typing, again, comes from a Greek word which means a print, like a fingerprint. So basically, we're trying to identify fingerprints in the genome. That's what we do. And that's most of the people do, and that's why personally I do. I don't do phylogeny. I do genotyping and genome comparison, therefore. So the genotyping, just like phylogeny, is based on sequences. So usually the same we use for phylogeny, but again, it is not phylogeny. It's only meant for identification and diversity analysis. There are different tools. You can do types, barcoding, MLST, which provide haplotypes. So specific set of sequence, okay? As soon as it changes from one single base, it's a different haplotype. However, because I saw that mistake very often, you don't do phylogeny with that, and especially you don't do phylogeny with MLSTs, okay? It is for typing only. So identification is an ID, and we compare the IDs, okay? So I insist on that. It's for comparing the different genotypes. It has no phylogenetic value. It has no classification value. So there is something, before going to the last one, but there is also, we see that very often, people use this kind of genes, and we'll see that later, that is completely not appropriate, to do classification, to give a species name, whatever. It's completely wrong. They are not meant for that. Absolutely not. And very often, and I must say something that we see so often in medical entomology, there is kind of race to give a species name. It's kind of competition, who will give the highest number of species name. And you have a species and the subspecies and the group and the complex and whatever. 99% of the time is completely artificial, they don't exist. So we have to be very careful with that. So the last definition, the last word which was in my title is phylogeography. Everybody understands what it is. It's the consecrated word. We know what It is, however, it's a wrong denomination. Because when we say phylogeography, it actually has nothing to do with phylogeny, okay? It's not phylogeography. What we are doing, we are just locating the different genotypes or haplotypes, so therefore the diversity, on the map and in time. So it's a spatial and temporal diffusion of this haplotype, of this diversity. It has absolutely nothing to do with ancestry and evolution, not at all phylogeny. So of course I will keep using the word phylogeography because it's the word we know, but it's not the most appropriate denomination because it's nothing to do with phylogeny. Okay, so now I'm going to address another kind of set of definition on the population, the species, etc., and the object of our study, because I talked about the tools and the approaches, but there is the object. We are working on, for instance, here Aedes, but it could be a virus, a bacterium, something else, doesn't matter. We are working on a biological object, and something that is living, something that is evolving. And there is one very important comment from Theodosius Dobzhansky, who is one of the father of evolution and species, definition of species, etc. He said something extremely important. Nothing in biology makes sense except in the light of evolution. We always should consider any kind of biological material under the light of evolution. It's extremely important. Otherwise, we don't understand what is going on. And it's true in our work. We're talking about phylogeny or diversity. And this is talking about evolution. And talking about evolution is talking about mutations. It's we saw that before. And I want to introduce, I don't know if you are familiar with, a very famous paradox, which is the Red Queen paradox. It's coming from Alice in Wonderland. And at one time in the book, in the story, the Red Queen is carrying Alice by the hand. And Alice is asking that, what's going on? Why are we moving backward? And the Red Queen said, we are not moving backward. It is the landscape which is moving forward. And then what happens is that the Queen is making Alice to run faster and faster and faster, just to stay at the very same place, very same position. And this is what we call the Red Queen paradox, and is extremely relevant in biology and in the kind of study we are doing. What is the morality of this story? It is that we live in an endlessly evolving environment, which is true, whatever. For instance, today we're talking about climate change. But our environment is always evolving since the beginning of time. So this is forcing us to adapt, to stay alive as a species. Okay, we must adapt to keep our position, to stay at the same place. So in biology, this is true, and it's true through selection and evolution, because evolution and life, they do not accept immobility. If we don't move, if we don't adapt, we disappear. We become dinosaurs. We just disappear. So, so genome plasticity is a key concept. And this is exactly what we are going to analyze when we do our diversity analysis, etc. We are addressing the genome plasticity. Okay, so let me move again to another series of concepts on species, population, and metapopulations before going to Aedes aegypti and albopictus themselves. Because this is very important in the way we analyze things and in the way we consider, understand, and interpret the results we have. So again, I'm going to cite Theodosius Dobzhansky, who gave a very good definition of a species. What is a species? A species is the result of the evolutionary process from which several sets, which were previously in an intersecting relationship, real or potential, separate into at least two distinct sets between which there can no longer be any intersecting. That's exactly the definition of a species. And it's very important because this definition is not I would say, the morphological definition. And we're going to go over that. What is very important here, and is the key issue for our kind of work, is that it gives the species a dynamic evolution. A species is moving, and you see, is made of different sets, and that we are going to see. For instance, if I try to illustrate what Dobzhansky said, and illustrate the point that we are talking about a dynamic thing compared to very often, especially in morphology or in classification, a species is a static thing. And that is a problem. Basically, what is a species? You have in all organisms different combination of genes, genomes, etc. Many genes. Most of the genes are shared between different species. The deeper in the evolution, the more they are shared. Some genes are present in all animals, some in all vertebrates, some in all mammals, etc. So the more you go up in this, the more specific you get. These genes here at this level are shared maybe by all the species because they are at the core and really the common ground. And the more you get specific. So usually, how do we define a species? We define a species through morphological traits based on color, on morphological features. There are feathers with color, the shape. So these traits that define the species in a morphological way is a very number of genes, very small number of genes. Only few genes correspond to that. And that is the characterization of the species, the expression of these genes, the phenotype they give makes that, well, we can say all the specimens, all the individuals who show these specific traits, all of them, not one, but all of them belong to the same species. And I decide to call the species A, B, Z, X32, whatever, or Aedes aegypti. I give a name, okay? But what we have to realize is this is very artificial in terms of dynamic biology and evolution. It is just our perception. And what we do is basically we take this very narrow range of traits and we put everything in a box with a sticker on it, and we put a name. And we work on this box and say, okay, this box is different from that box because it is not the same species, because they don't have the same morphological traits. So two different boxes, two different stickers, two different names, okay? But that's fine as long as it comes to classification. It's perfect. For classification, it's perfect. We have to be careful because sometimes this is completely artificial. It does not really encompass the biological reality. It is just our perception. So, but when it comes to have clear, well-defined morphological trait to classify, is perfect. But it's perfect for classification and only for classification. When it comes to other kind of analysis, it's wrong. Okay, because a species is not static. A species is something dynamic with only few genes being specific between two species, most of the genome being shared. So we have to consider that when we do the kind of analysis we are working on and the kind of genes we select for the analysis. If we select a genes which is very deep here in the blue, but we will not make the difference between any species because the gene is present in all of them. Okay, so it's very important to understand that. So now I'm moving to something a bit different, but it's exactly, I'm going to illustrate the definition given by Dobzhansky, by Tadeusz Dobzhansky on the species. What did he say? He said is a species is something comprising different sets that are more or less in interaction, and they may lose this interaction and become separated so much that there is no more interaction. So basically, that's the main difference between the species in morphological traits. So we can consider Aedes aegypti as a species is one block, and all individuals in that are the same. This is not the right approach. Again, this may be good for classification, not at all for what we want to do, because we must acknowledge and see and understand that a species is composite, as defined by Dobzhansky. What we call a species is actually a meta-population. So a population of populations, a group of different populations having different genetic characteristics and different evolutionary trends. Okay, so basically, when you start from the beginning, you have the population, so and the male, female, and sometimes a mutation happens in one individual. Mutation happens, so the genome is no longer the same, and this will be shared in the progenitor. You know that very well. Okay, there's nothing new about that, and this will translate in an evolution from an initial population into the evolution with time of different population. And these different populations themselves subdivide in other populations. That's exactly what we're studying when we do the diversity analysis or the phylogeography. We are working on this subset population. We are working on separate population, and that is very important. And it has a very important aspect also, is that many important traits, many phenotypic traits are linked not to the species, but to the population. For instance, it's one example, but there could be plenty of different examples. One example, for instance, is the vector competence. Okay, some population will be competent for carrying the vector, the virus X. Okay, whereas other population from the very same species will not be able to carry this virus because they are not competent or very poorly competent. So the competence, the capacity to carry the dengue virus, for instance, does not depend on the species because they are all Aedes aegypti. It depends on the population. At that level, the species is not representative. That is why it is important to work on the populations, not on the species. And that's what we are doing. So I said earlier, yes, okay, now we are going into our results and on Aedes aegypti and albopictus. Okay, it's important to work on the population. It's important to compare the different populations, compare the different haplotypes, genotypes, etc. We mentioned, I said before that phylogeny or any kind of analysis you're using the phylogenetic tools, and you're working on diversity. You don't work on the global genome on the species. You work on specific genes. So the point is, which genes are we using? What are we working on, actually? So when you look at the literature, what is done everywhere, and it's important if you want to compare, you have to work with the genes that everybody is working on. Otherwise, you don't get information. If you are the only one to use one gene, you get information only from yourself, and you cannot compare with others. So in that case, very often, we have three basically targets for this kind of analysis. One gene very often used, and I must say is the one which is used the most often. So in other words, it's the one for which you find the highest number of information in databases, is the CO1 or COX1 or cytochrome C oxidase subunit 1. What is important to understand, and we're going to see that, this is a bacterial gene because it's into a mitochondrion. So there is no recombination, there is no crossing. And what we study when we analyze this gene is we are studying maternal lineages. I'm going to talk about that in 2 minutes. The other target we are using is ITS2, which is the internal transcribed spacer 2, which is a ribosomal sequence, a nuclear sequence. Sorry, I didn't write it here, but it's very important. It's a nuclear sequence present in the nucleus of the cell, okay? And so the sequence are coming from both parents. And this is used exclusively for phylogeny. It is basically useless for typing. Because it's very highly conserved, so you don't see much difference. On the other hand, if you see a difference with ITS2, it means that you are really dealing with 2 different species. But otherwise, most of the time you see nothing. If you are within the same species, the same metapopulation, you will see nothing. It's too much conserved. It's not for diversity. It's mostly made for phylogeny. And there is a lot less information in database than for a COX1. The last tool which is used is the microsatellite, which is completely different. It's not a gene, it's not a coding sequence. It's a series of repeated sequence. And it's very accurate, too much accurate. It's very good for individual genotyping, for genetic mapping as well. But it's too much accurate because it works at the level of the individual. For the kind of work we want to do, it's too much accurate. We cannot get anything. It's too much, we cannot make a good analysis of it. So basically, we are working with 2 sequences, COX1 or ITS2. So let's go back, I said, between the origin. And this is extremely important because I saw many, many times a lot of confusion between the 2. And people wanted to compare the results, for instance, the analysis they got with COX1 with analysis they get with ITS2. Well, most of the time it's different. And they say, oh, there's something wrong, it's different. No, it's normal. The exception is that it tells the same thing. Because there are 2 different genes. As I said, COX1 is from the mitochondrion, which is a bacterium that developed symbiosis with the eukaryotic cell. So the genome is within the bacterium, is replicating like a bacterial genome. There is no recombination, no integration from the other parent. And since it is present in the cytoplasm, it is only acquired through the mother, okay, through the cell. The father participates to the production through the spermatozoids, so not through the cell. The mother is providing the cell. The father is providing only the chromosomes. So the father has nothing to do with the mitochondrial DNA, only the mother. So when we do that, we work only on maternal lineages. However, when we work on nuclear targets, like ITS2, it's located in the nucleus, the nuclear DNA. The material is coming 50% from the mother, 50% from the father. So you have a lot of recombination, a lot of integration, a lot of mixture, and a lot more diversity. Except, as I said, for ITS2, which again is not a coding sequence. It's a very special, highly conserved spacer, and it is used actually again for phylogeny. So really important to keep in mind that we have these different targets. And for the kind of work we want to do, but I would say that basically none of the targets we use are actually completely relevant. And that's the problem. CO1 is a bacterial DNA maternally inherited gene. It's haploid, it's not diploid. So it's not at all relevant for population genetics. For population genetics, you must work on diploid genomes with origins from both the father and the mother, so on the nuclear DNA, so not COX-1. ITS2 is meant really for phylogeny. But what we need to do is genotyping. So it's not fully relevant either, okay? And on top of that, none of these genes we are going to use for this work correspond to the genes involved in the morphological traits used to define the species. None of these genes is coding for in the ITS2 is not even a gene. It is not coding for the color of the skin, for the shape, for the feathers, for the, it's not coding for anything like that. So it doesn't get involved in the species definition. So you cannot correlate those genes with the species. So it's wrong for classification. And but they don't correspond ever to physiological traits like resistance, vector competence, etc. For instance, you see a map of resistance of mosquitoes in Indonesia to chemicals. The genes we have here, if we analyze them, it's totally useless for understanding the dynamic of resistance because they are not related to the genes involved in resistance. The only thing that is very important for us in the terms of analyzing the diversity is that the COX-1 or CO1 can distinguish the different species. And you will see in the last set of examples that is very useful to correct mistakes. So basically, and it's very important, all we can do now when we do this kind of work, we are more or less, not always, but very often, basically restricted to characterize maternal lineages. That's it, not more than that. Okay, so again, I said maternal lineages is that the mitochondrial DNA is coming from the mothers. So we have maternal lineages, so we will compare different maternal lineages. So we will say that, okay, this population is coming from these maternal lineages, this one has different maternal lineages. That's basically what we can say. Whereas the nuclear DNA is inherited from a lot from different ancestors, so it's extremely mixed. It's basically impossible to really analyze with origin. ITS2 is different because it's highly conserved. So let's go back to Aedes aegypti and albopictus, okay, which is after all the real story we are interested in here. Okay, so let's first say some work we did during the PhD of Tribo Ambargagito, who is working in Salatiga in the former IVRCRD, no, he changed the name. So Tribo was my former PhD student, so he very brilliantly defended his PhD, and now he's Dr. Tribo Ambargagito, and did an excellent PhD, excellent work on different mosquitoes and viruses. One part of his work was the genotyping of Aedes aegypti and Aedes albopictus in Indonesia. So we collected samples all over Indonesia in 25 provinces and in 43 locations, okay? And we analyzed them, okay? Separately, of course. Yes, this was done under the help and funding of the Vectora project from the Ministry of Health in Indonesia. So let's talk about aegypti, Aedes aegypti. What did we find? Again, all over Indonesia. It's not one place, it's all over Indonesia. And we analyzed all these sequences with the same tools, of course, at the same time. What did we find? We found that, well, Aedes aegypti was extremely and surprisingly homogeneous all over Indonesia. All samples belong to the same maternal lineages. Remember, it's the CO1. Huh? So same maternal lineage. That's only one, okay? And not only that, almost all aegypti samples collected in the study, so you see 87%, belong to the only one population. That one in blue here, which has itself separated in two haplotypes, H1 and H4, which were by far the most represented all over Indonesia and all over the place, and a lot in Java, actually. And importantly, this population was completely different to that which has been described in 2013 in the publications, in the literature. They describe Aedes aegypti, but it was genetically not related. It was a different population, a different maternal lineage. So, so something happened between 2013 and 2016 when we started our sampling, because we changed the maternal lineage. Okay. What about albopictus? Exact same thing. Exact same thing. We found, well, we had a lot less samples here. So we found 3 haplotypes, 1, 2, and 3. And these were completely different what has been described in 2013. So the paper is 2017, but it comes from earlier. And there was haplogroups defined by Battaglia et al. In 2014. And this work done by Mena et al. In Indonesia matched perfectly these haplogroups, but and most of them belonging to 2 specific haplogroups. However, in our work, the sequence we got from the samples we collected, these 3 haplotypes here, they did not match at all with any of the haplogroups defined by Battaglia, and they differ also from what has been described previously. It looked like we had somehow the same story, different maternal lineage all over Indonesia. However, I must say that here the number of samples is a lot smaller, so the study is less significant than with aegypti. So that first story. So we did also the phylogeography. I want to give, I leave this question under suspense for the moment. We're going back in a few minutes. Now, phylogeography, again, it's not phylogeography, it's mapping the haplotypes. I just want to tell you about some previous work we did, not in Indonesia, but in Vietnam, okay? And in Vietnam, we did the very same thing we did with tribo in Indonesia. We collected mosquitoes, aegypti and albopictus, so here aegypti. We collected mosquitoes in different provinces in Vietnam. And we sequenced and we analyzed the CO1 sequences, the maternal lineages. And what we found out is that the lineages we found in these Vietnamese provinces could be found exactly the same in many places around the world. Exactly the same haplotypes was present in other places. That's a very wide distribution then. Whoa. And if we do the same kind of analysis with albopictus this time, we got exactly the same result, except that the range of distribution was smaller, okay? Mostly because also we have to consider historically that Aedes aegypti, which is coming from Africa, has been spread worldwide since the 18th century, basically, whereas albopictus, which is coming from Southeast Asia, has been spread worldwide only very recently in the last 30 or 40 years. So, and, but same story, the phylogeography shows that it is present in many places around the world, exact same haplotypes. Oops, I'm sorry. And if we do something else, we look at continental Southeast Asia and mostly Thailand, Cambodia, Vietnam, which are the countries where we have the most information actually. So this is not the mosquitoes. This is the dengue virus, the 4 dengue virus. But what we found out is that some dengue virus are found only in one place, in one country, only in Thailand or only in Vietnam or only in Cambodia, and they don't move. And other dengue virus, other virus lineages, they are transborder lineages. They move between different countries. And we have to consider that a virus does not move. The virus moves because we move it. Okay, and from the analysis, we have been able to exclude the human mobility of the virus. So the only way to explain the mobility of these viruses between the different countries is because they are moved, they are carried by mosquitoes. Mosquitoes who move from Thailand to Cambodia to Vietnam or the other way, mostly is bound. Mostly is from Thailand to Vietnam. Very rarely the other way. And but mosquitoes like Aedes, they do maybe a few hundred meters, 200-300 meters maximum. So they cannot fly from Thailand to Vietnam. So if they move, it's because they are transported by humans. So basically, you have some kind of mosquitoes who are living in an environment which corresponds to something that is moved by the humans. And these mosquitoes carry some viruses, and these viruses move also. Okay, it's what we see exactly when we look at the virus lineages, again, we see different lineages, and all the lineages, the viruses which move are all monophyletic. Basically, they correspond to the progeny of the daughter, if you want, or the previous virus. Unrelated virus, like this one, for instance, will not move. So we are trying to see what happens now at the scale of the whole planet, okay? And we are now conducting a work on the worldwide genotyping of Aedes aegypti and Aedes albopictus. So this is work I'm doing in Indonesia here, of course, with Tribowo Ambardarjito, but also with my colleague and friends, Professor Tribaskoro Tunggul Satoto at Gajah Mada. Our master student, Gabriela Stéphanie Kuzuma, who is working on this project, and in France with 2 colleagues, Laurent Gavotte and Sylvain Terry. And what we did, we took all the NCBI sequences we could find of COX1 or CO1 and ITS2 for both aegypti and albopictus. We collected everything we could, and then we cleaned that. We retained only the full-length sequence, okay? And we eliminated those who were too short, etc. So we kept only we eliminated the sequences with obviously many mistakes, etc. So, and we have a whole set of basically more than 2,000 sequences to work with. So a lot more for CO1 than for ITS2. And we are doing genotyping and phylogeography. You see, I don't say we are doing phylogeny, we don't. And I don't say we did, because we're doing it at the moment. What I'm going to show you, the trees I'm going to show you, I got them yesterday. So I'm going to show you some very preliminary results to give you an idea of what happens and the problems we can encounter. And we haven't done yet the phylogeography. We are going to do it. We just did some preliminary genotyping and only on aegypti. We haven't checked yet the albopictus sequences. So what I'm going to show you is preliminary and only aegypti. So, and it's very interesting. First of all, so you see what we get? We get the husband. When you see the CO1 from aegypti full length, we get that. See, what is this? We have a whole bunch of sequences here, all the same, by the way, exactly all the same, which come from China. And you see, and here above you have some other sequences we use for reference of the Aedes, sorry. For instance, here, the illustration of problems we can encounter. Here we have a sequence of a culex, which is registered in the database as a culex, but which is obviously an Aedes, and something related to Aedes gallosi. So mistakes happen when people name. They make a mistake in identifying the species, and then the sequence is associated to a wrong species. And you have to be very careful with that. You will see examples again. So, and all these sequences coming from China, and all of them from 2016, are actually the same. They could have put only one sequence, it's the same thing, because it's maybe 1,000 times or something like that, the same thing. But it's a whole bunch, it's a large number of sequences which are the same, which impact the database and impact the analysis we can do. And then we have another series of sequences here in black, and you see they are not related. Okay, the green, the Chinese, and the black here, very, they are distant, it's not the same. Okay, and these one are Aedes aegypti. Okay, and you see, except for they are basic, except for a few exceptions, they are exactly the same. And you see, it's impossible to say anything about it. We cannot analyze that. It's impossible to analyze. So we tried to understand what was going on, and we were very puzzled by these Chinese sequences. And we thought, wow, these are not aegypti. It's a problem. It's another mosquito. They made a mistake, and we tried to investigate, and actually we found out it is aegypti. They didn't do a mistake. It is aegypti. However, what they did, they didn't sequence the same region as all the others. Yes, it is CO1, and it's labeled as CO1 in the database, but there's only a small part which is CO1 and overlaps with the other. The rest is different. So when you do the phylogeny or the diversity, but the phylogenetic analysis, but on the genotyping here, the software is working on the common. As I said, it doesn't take in there. So it's just focuses on the part which is exactly the same. And this part, which is exactly the same, is very small, because a large part of most of the sequence here is outside the COX-1. So the analysis is not done on the full gene, is done only on a small part of the gene. So basically, it's very stochastic, and it's not significant. So these Chinese sequences here, which are present in the databases in NCBI as Aedes aegypti COX-1, are actually only very partially COX-1, and when you include them into the analysis, it breaks the whole analysis. You cannot do anything. So what we did, we redid the whole analysis after having excluded all the Chinese sequences. So we get rid of all these green sequences, and we do it again. What do we see here? We see something completely different, And again, it's preliminary. So we have some things like here, something very interesting. For instance, this is in Madagascar, but we have something similar here in El Salvador, in Latin America, a group of sequences of Aedes aegypti, which is only in Madagascar and which is quite diverse, but only in Madagascar. Why, we don't know yet, huh? But only in Madagascar. However, when you look at everything else, it's extremely similar. The bootstrap are very small, so not significant. It's very small. So again, I want to illustrate some example. For instance, we used also some culex to root the tree, and we found out that some strains identified as Aedes aegypti actually were not Aedes aegypti, they were culex pipiens. So you see, mistakes happened. Same thing here with the albopictus. We found that some strains identified, or some mosquitoes identified as aegypti were actually albopictus. So we have to be very careful when naming, because it's a lot better to name after the sequence has been analyzed when you have something clear than before, because mistake will happen. And these sequences, for instance, are now considered aegypti, but they are not. They are culex, actually. And what we see here is basically we have two populations here of Aedes aegypti, very closely related, very, very closely related, but two populations, or you cannot see it here, but which spread, both of them, all over the world, all over the world. Okay, so I'm going to summarize. What did we find? We found that there was misidentifications, problems with the region sequence, like the Chinese. So this is something we have to be very careful about, that distort the whole analysis at the first glance. And it's only a first glance because, as I said, we are working on it. I just wanted to give you the latest result. It seems that we have only two very closely related populations of Aedes aegypti circulating around the whole world. It's the same, okay? So, and these are again maternal lineages. So what happens is that we need to now work, the work we are going to do now, to work on the phylogeography. See these two populations, where did they go? Which country at what time, and try to correct that with, for instance, some maritime routes for the diffusion of mosquitoes by boats, etc. This is the work we are going to do now. We haven't done it yet. But for the moment, what we can already say is that there is not a whole series of different populations, haplotypes of mosquitoes around the world. No, it's the very same. Maximum two. But these two very closely related, which circulate all around the world. Now we need to understand how they circulate, what are the routes, etc. Again, these are maternal lineages, because it's basically the only thing we can do. So we did the same with ITS2, but basically ITS2, as expected, gets basically no information because it is too conserved and it's not meant for genotyping anyway. It's meant for phylogeny. And a lot less sequences are available. And on top of that, most of the time, POX1 and ITS2 sequences in the database are not coming from the same specimen. So we don't have the same coverage in terms of geography, of mosquito specimen, etc. So it's basically impossible to compare. And on top of that, it doesn't give any information, actually. So that brings me to the conclusion. Something very important is that sampling and sequencing must be standardized. We have to use the same genes, the same sequences, to use different genes from the same mosquitoes when you do that, to deposit that in the same database with the same tools, etc., to have something well standardized. And that's exactly one of the work we are doing, also with my colleague, Tungul Satoto in FDM. We are developing right now exactly that kind of tool, a map-driven and GPS-driven integrated database to standardize, collect, standardize everything from the time of sampling the mosquitoes to the time of analyzing the sequences. That is the future work we are developing now. And with this, I will end this presentation. I will thank you very much for your attention.
Okay. Thank you very much for excellent, detailed, and clearly presentation.
Thank you.
Yeah. And now we are going to open discussion. Someone who want to deliver a question could raise hand or write in the chat. Please. Any question? Muhammad, do you want to deliver question?
Yes. Yes, yeah, yes, of course.
Yeah, please.
So
Yeah.
I believe must more or less answered that question during the talk, actually. So right now that we see is that in Indonesia, but elsewhere is the same. Population are very homogeneous.
Okay. Yeah.
So there is not much diversity. However, you see, it happened in Madagascar. It happened in El Salvador. You have some pockets where the mosquitoes become more diverse in one place. We don't know why. We don't understand why yet. We just saw that. It's very interesting to try to understand why. But so this could also happen in Indonesia. And Indonesia is an archipelago, a huge archipelago, with plenty of islands, with different environment, different climates, etc. So this could very well happen.
Okay.
In our work, our experience in Indonesia, it's basically all the same.
Oh, okay.
At least for the moment. That's what we saw. But again, what happened in Madagascar, what happened in Salvador, why not? Could happen also in Indonesia. It would be great if we could find something like this and try to understand what is going on. Huh?
Okay.
Yes. And I don't remember the rest of the question. Can you show it again?
I think, yeah, someone, Muhammad.
Yeah. Yes.
Please.
I see it here. Let me open it.
Yeah.
Okay. Can phylogeny explain which genes? No. As I said, first of all, we don't do phylogeny at all. So we do diversity, actually. And nobody does phylogeny. and again, the genes used for this kind of analysis have nothing to do with the vector competence. We are not working on the genes involved in vector competence, and we don't know. We don't know what are the different genes involved in vector competence. We don't even know which. So it's not possible to work on those genes. We cannot. We cannot. It is, let's put it that way. It is not through that approach that we can solve the problem of dengue virus, of the spread of dengue virus. We have to use another approach. We are working on that also, but it's a different topic. It's something different. I didn't address that.
Yeah. Okay. So actually, I would like to ask you why
we have to forget about the genes and everything. We have to focus on the management of mosquito management at the level of the community.
Okay. Yeah. Actually, I would like
this kind of work will not give any information for that. And then, is it possible to stop the spread of the disease? I don't think so. There are some work in transgenic mosquitoes, but it's just a lab work.
No.
As soon as you put them in the wild, I worked a lot on transgenic plants. Transgenics are very fragile organisms. But transgenic doesn't stand long in the wild. It will be replaced by the wild populations anyway. So my personal answer to that is no. It's a nice laboratory, is a tool, is a work in the lab. It's interesting from technical standpoints, etc. On operational standpoint, personally, no, I don't believe it.
Okay. Next question, please.
Okay. I have a question.
Yeah.
Thank you, Prof. Roger, for his presentation. And also extend to my thanks to Prof. Indang Rumoni and Dean Prof. Ido Duroha. And I also thank the participant. So actually, this topic is very hot and very important. As you know that Aedes aegypti is most important vector of many arboviruses. And Southeast Asia is the endemic area that put a lot of burden on the public health, like dengue virus and chikungunya virus are a study of Aedes aegypti, the genetic diversity or the population genetic of Aedes aegypti is very important, and using of the multiple markers is very helpful, like CO1, IST, and microsatellites is very important to understand the population genetics of these vectors. And I also, in the last 3 years, we did also, in my PhD research, I did study on Aedes aegypti based on the CO1 gene in a very small area in Central Java and West Java in Jakarta and Bandung areas. So we found also, our results also is same as your study you did previously. So we found that Aedes aegypti is from same material, and we found a lot of haplotypes and also haplogroups. So these haplogroups, some of them were similar to that were found in Thailand, some were found in Africa, some were found in New World like Brazil and so on. So, but as you say, that the problem of CO1 is not give us the bigger picture of the population genetic or the genetic diversity of these species. So your study is very comprehensive and very good as you use the combination between the CO1 and IST. So I have many questions, but I will briefly, one of the questions is how as you said that the CO1 gene is maternally inherited, and the bacteria, the symbiont, the bacteria that found in the mitochondria, how it affects the evolution of this, the evolution of the mitochondrial DNA. And also some study found that there is some pseudogenes during the DNA modification, some of the mitochondrial DNA or the CO1 gene is integrated or transferred to nucleus. And how we know this, the genes that we are amplifying is the original mitochondrial DNA or the pseudogenes. And also, I have third question that in your study, did you found that one of the lineages, as you know that Aedes aegypti, there is 2 lineages, East Africa, and there is some also the West Africa or the New World lineage. So in Indonesia, do you found 2 lineages or 1 lineage or like this? And also I have another question. So what's the implication of these studies? Everyone do population genetic diversity, phylogeny, phylogeography studies. What's the implication of these studies on the control of the vector and their implication on the mitigation of the spread of the arboviral diseases? So thank you so much.
Okay, thank you. So I will start, sorry, by the last question, then you will need to remind me the other questions because there are several questions.
Yeah.
So the last one, if I understood well, what is the importance of this genotyping for the usefulness, I would say, of the genotyping for the control of the arbovirus diseases, right?
Yeah, right.
That is the last question, right?
Yeah, that's the last question.
Okay, so my answer to that is very simple. It has no use. We cannot, from this information on genotyping, etc., it doesn't help at all controlling the vectors and preventing the spread of the disease. Doesn't help.
Yeah.
Because the gene, especially COX-1, has absolutely nothing to do with the vector competence because it is not a nuclear gene. It is a mitochondrial gene. It's a bacterial gene. in the mitochondrion is not a nuclear genes, or the genes involved in vector competence, etc., are present in the nucleus. They are nuclear genes. COX-1 is mitochondrial genes. It has nothing to do with that, no relationship at all. So the information we get from COX-1 does not help at all. It is basically useless for controlling the virus and controlling the mosquitoes. We have to have other approach.
Yes.
This only tells us on the structure of the population and, to be clear, on the structure of the maternal lineages. We know that, for instance, 1 or 2 lineages are circulating. That's all we know, and that's all we can know. We cannot know more than that, actually. It doesn't tell us anything on how to control the mosquitoes and how to prevent the spread of the virus. We have, but it's very important information. We know that it is useless, so we have to think differently. Controlling the virus and controlling the mosquitoes, it's not a matter of genetics, it's not a matter of phylogeny, whatever. It's a matter of behavior. It's a matter of control. It's a matter of population awareness and train the population to eliminate the mosquitoes at the level of the community, etc. It's a completely different issue. And it's the only way we can prevent the spread of the disease is by lowering the frequency of the infectious bite. So therefore, by controlling the mosquitoes. And we don't need to know the species, we don't need to know the population, doesn't matter. What we need to know is where they live, what are the breeding sites, and we know that, and eliminate those. That's it. Really, that's the only way to control. Then the kind of work we're doing is important to understand the dynamic, not for controlling.
Okay.
And
it is great.
I forgot the previous question, so maybe if you can write them. And,
yeah, my previous question:
one by
how the bacteria that we found the effect found in the mitochondria affect the inheritance or the evolution of the CO1 gene?
How, I didn't understand exactly. Could you
repeat again?
Sorry?
Yeah, my question is that there is the symbiont bacteria or the bacteria albopictus were found in the mitochondria of Aedes aegypti. Yeah. So how these bacteria affect the evolution or the inheritance of mitochondrial gene like CO1? The other question is during the DNA modification or DNA regulation, there is some part of the CO1 gene integrated or transferred to the nucleus of the organism. So nucleus. So how you know that the CO1 gene you are studying is it from the mitochondrial or from the nucleus?
No, CO1 is not from there is no CO1 in the nucleus. There is no transfer. CO1 is purely mitochondrial. So let's the mitochondrion is a bacterium that develops a symbiosis with the eukaryotic cells, basically maybe before, but at least at the time of what we call the big oxidation. So basically 2.4 billion years ago. Because if you just to make some short parentheses about that, the eukaryotic cell has never been able to solve the problem of conversion of energy. So all eukaryotic organisms, including us, we are all symbionts.
Yeah.
Because we can breathe, because bacteria is doing that for us, specifically the mitochondria. So there is bacteria which are associated with eukaryotic cells, and then they become a part of the cell itself. So it's bacteria which degrade the oxygen, because oxygen is a poison, and they degrade oxygen, and they make energy out of it. So that's the role of mitochondria, is to provide the energy also. But even for the food, we digest food and we get our energy. We don't digest. For instance, ourselves, we don't digest. We just transport the nutriment. It's the bacteria that digest the nutriment for us, the food for us. So again, we are symbiont. So the bacteria has been associated with the eukaryotes from the very, very beginning, 2.4 million years ago, and maybe before. So they are separate in a karyotic cell like us, but the mosquitoes, you have two kinds of DNA, the nuclear and the mitochondrial. They don't exchange. There is no exchange between the two. And the COX-1 is exclusively mitochondrial. So it's bacterial and maternally inherited. It has nothing to do with the nuclear genes. So there is no exchange, and there is no recombination in COX-1, and there is no involvement of any mitochondrial gene in the phenotypic traits linked to the mosquito, like vector competence, resistance to insecticide, color, shape, whatever. It's completely unrelated, totally unrelated. That is why I said that basically none of the targets we use are actually relevant for the question asked, because the real question for us behind all this work is how can we prevent the spread of the disease? How can we solve the problem of resistance to pesticides, for instance? That's the question we want to answer. And these kind of markers are completely unrelated to that. They cannot give any answer for that because they belong to a system which is completely different.
Yes, thank you so much.
Okay. Any other question from audience? If not, maybe from me, Professor Roger. As you mentioned that, you use the CO1 and ITS2 to construct the phylogeny and phylogeography. Yeah. Which one better between two primer that you are using? Or it is in different, yeah. Which one better when you use the CO1 compared to ITS2 to construct the phylogeny and phylogeography? It is any differences?
Okay. If you really want to do phylogeny, phylogeny, as I said at the beginning of my talk, to work on the real phylogeny, so do real phylogenetic analysis. So understand how the different characters, the different mutations moved from an ancestor to. You don't want to characterize the populations. You want to understand the history, the phylogeny. If you want to do phylogeny, you've got to use ITS2. Okay. Because ITS2 is meant for phylogeny.
Okay. Yeah.
Is really ITS2 is really something for phylogeny. Okay. So that's why doing genotyping with ITS2 doesn't really work because it's not meant for that. Okay. Oh. Now doing phylogeny with COX-1, real phylogeny again, not genotyping, real phylogeny with COX-1. You don't do a real phylogeny, you just characterize the maternal lineages. But you exclude then all the nuclear sequences if you do COX-1. So I would say if you really want to work on phylogeny, then the best target for you is ITS2.
Okay. Yes, thank you. Yes, actually, I would like to ask you about the vector competence and based on the gene, but you already explained that you don't know or you not in this area, right? So I would not continue in my question. Yes. Okay, thank you. And then any more question from audience? No? Yeah. Okay. So
Yes,
if there
I
is
have
no
another question, please. If
Another
you
question.
allow me.
I don't know where.
Yeah. Who it is?
Other question. I'm sorry. It's this time I just I want to ask some questions. Just it's related to terminology. I would like to know what is the divergence, and also the difference between the heteroplasmy and homoplasmy. These terms usually used in the population genetics of these aegypti.
So what was the first question? What is
the term? What's the term of the divergence? I mean, also you already yeah.
So here you are really working. So diverging. What is diverging? Right? Divergence.
Yeah. Yes. Divergence. Yeah.
It's the result. It's the result of evolution. I mean, you have accumulation and mutation, and the populations are population are diverging, okay, from a common ancestor. Okay, so they start from a common ancestor, and it's exactly what I showed you. Remember in the beginning when I showed the initial population and two population of Aedes aegypti. It's because in a given initial population. You have mutations occurring in different parts of the populations, and the population itself is again what we call a population. A species is actually a meta-population, so a population of population, and mutations occur, and not the same place. They don't have the same function. They give an advantage or not. Within the same species, the different populations are not under the same selective pressure. They are not in the same environment. So this is very diverse. And starting from the same ancestor, because of mutations, because of adaptation, evolution and adaptation to maybe different environment, the populations will evolve differently. Through time, they will evolve and adapt differently through time. So therefore, from the single population initial, they will diverge. They will diverge to a point, and that's the definition given by Dobzhansky, up to a point where the difference is such, okay, they accumulated so much differences that they cannot interact anymore, and they became two different species. The process of speciation, but it's a permanent process everywhere. It's a continuous process. It's true for everything. It's true for every living organism. As I said, the world is moving all the time. The environment is changing all the time. So we, not as individual, but as a species, as a meta-population, we are evolving. We are adapting. So we change all the time. Humans today are not the same as humans 20,000 years ago. We evolved and we keep evolving, and it's true also for mosquitoes and for anything else. And that is the divergence, is because we start from a single point, but because we are exposed to different conditions and because mutation occur, with time, through time, we are taking two different paths, two different roads, and then we diverge. And this divergence can lead to the occurrence of two different species. So basically, even if we come from the same initial population, after a while, there is so much divergence, so much difference that the two populations cannot breed anymore. And that comes also to the definition of a species. A species is also defined by the reproductive isolation. individuals from two different species cannot reproduce. Or if they reproduce with, for species which are quite close, the progeny is sterile. Like tigers or lion or donkey and horse, they can reproduce, but the progeny is sterile. So they diverged so much that they became two different species. I hope I understood your question. I think I understood that was your question. Tell me if I'm right.
It's okay, Muhammad.
Yes.
Yeah, it's enough.
Yeah,
Yeah,
it's okay. It's okay. Yeah, it's okay.
no more question again.
No more question.
Okay. And
For
other
me.
people? Is there any question? Okay. If there is no question again, and then we will finish for this presentation and discussion. And then thank you very much for Professor Roger Frutos for the nice presentation, for the good presentation, yeah, and it is useful for us to understand about the phylogeny and phylogeography. And the time now I give to MC, Masters now. The time is for you.
Okay, thank you, Prof. Endang and Prof. Roger. This time I would like to call the speaker, Professor Roger Frutos, to receive the certificate. Kindly please, the Dean, Dr. Dwi Nugroho Wibowo MS, to pass on the certificate.
Okay, thank you, Mas Pino. It is an honor for me and the Faculty of Biology, Jenderal Sudirman University, Purwokerto. You are pleased to come and share your knowledge with us about the diversity, phylogeny, and phylogeography of Aedes aegypti and Aedes albopictus in the second series of webinar. I think we agree your presentation this afternoon is very interesting topic. Once again, Prof. Roger, thank you for being interested in our program and for being willing to share your information, experience, and knowledge with us, which is certainly very useful for all of us. Please accept the certificate as a token of our gratitude at the Faculty of Biology for you. I hope to see you again at another event. Please, Prof. Roger.
Well, thank you very much, Professor Dwi Nugroho, for this certificate. It's for me a pleasure and honor to receive the certificate. And thank you again very much to you, to Professor Endang, to the faculty and university for the invitation and the opportunity to give this presentation and discuss with you this issue, which is really important on analyzing the different population of mosquitoes.
Okay.
Okay,
so thank you very much.
You're welcome, Prof.
Thank you. Terima kasih.
Yeah, you are welcome.
Bye.
Okay, thank you very much, Dr. Dwi Nugroho Wibowo MS. Okay, for everyone here, the presence for these events have share in chat column. Yeah, you can present at the chat column. Okay, ladies and gentlemen, we finally come to the end of this event. Let us close the webinar by reciting Hamdalah, Hamdulillah, Rabbil Alamin. Okay, thank you so much for your attention.
Thank you very much, Prof.
Thank you very much.
Thank you very much.
Thank you.
Thank you. Bye. Thank you.
Very nice time.
See you, Prof. Roger.
Bye. Bye.
Yeah.
Bye. Thank you.
Okay.
Muhammad
I want to say sorry for
terima kasih. Terima kasih.
Yes, thank you very much for everyone here. Bila taufiq wa hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Have a good day and see you.
See you again next time. Bye. I'm leaving.
Sorry, sir. Before we close the room, let's take some picture for the last documentation. Please open camera for everyone here. Please, the last documentation. for the last documentation. Okay. Okay. 1, 2, 3. Okay, please. Another participant can open your camera. Okay, wait a minute. Okay, the last 1, 2, 3. Okay, thank you
Okay,
very
thank
much.
you.
See you. Have a good day.
Thank you.
Tapi mungkin ada di YouTube ya.
Thank you for everyone who
Yeah.
participate with
See you on the
this.
next series and
Yes.
biology. Yeah.
Alhamdulillahirrahmanirrahim.
Matur nuwun Mas Una, Mas Ino, Mas Ino, Prof Endang.
Terima kasih, Cinle. Selamat, selamat, Prof Endang, semuanya. Terima kasih semuanya.
Pamit ya, Mas Ino, terima kasih, Prof Sorta.
Nggih, matur nuwun Pak Dekan.
Pamit.
Izin ditutup nggih Pak Dekan.
Monggo.
Izin, Lab.